So today's session is focused on soil health science, so science behind soil health practices. Today we have Nathan Stacy and Shannon Kapalasi with OSU Extension. The three key outcomes we have for today is to have the group build a shared baseline understanding of soil chemical, physical, and biological health. I heard that kind of baseline understanding was something that you all were looking forward to today. The next outcome is to connect around soil health principles and or connect the soil health principles to the practices across different cropping systems. So we can talk about the principles and practices in general, but what does it look like on the ground across the state where you all are serving very diverse regions and landscapes? Lastly, it sounded like this was a really key, interesting point for you all is to learn more about some other soil science programs and specifically the database that Shannon is working on and how to get involved. So we will, Shannon will share more about that, answer lots of questions. And as a reminder, we will have another resource training. So our last training session in the series is just focused on soil health resources. So we'll get to dive more deep into that again in December. And just a note on today's session, OrCAN recognizes that soil health practices and principles are not new. Native communities have long understood that soil, plants, water, animals, and people are interconnected. So we do honor the historical, spiritual, and ecological teachings that inform these land care traditions, especially those held by Black, Indigenous, Latine, and other farmers of color. And so this training does focus on more of a Western science perspective, and we'll make sure to follow up with resources highlighting both the resources shared today and Indigenous and ancestral soil health practices, which you can also find both on OrCAN's resource pages. All right, Shannon and Nate, I'll pass it off to you all.
All right. I guess we'll do a little introduction here. I think most of you have met me or heard me speak at least a time or two. I'm really excited to have a group of soil nerds where I can nerd out on everything I've been working on for the last 15 years as well. Just as Sage was saying, I have a lot of information on the database available, but I put just a small amount in this presentation. There are also some really nerdy topics that I have slides for in the presentation after the planned topics. So as we're going through, if you're like, yeah, we know this, let's get to like some kind of interesting, cool little things that we might not know about, feel free to let us know. Nate and I want this to be really conversational. He's going to chime in whenever possible, maybe any kind of clarification. And I want you all to feel the same way. Like I want to hear your questions, hear your pushback. If there's anything you want to challenge, do it. If there's anything, it's like, well, that's not quite what I've seen. Let's talk about it because that's what we're here for. This is ongoing research as we're trying to understand how biology influences everything we do in agriculture. There's a lot to learn, and I've studied it for a long time. And I also know that people who are out there in the field have had different experiences, and so I want to hear your perspective as much as possible. As far as introduction and going back to that, I am the soil health extension specialist within the organic ag program at OSU. We serve all producers regardless of whether they're organic or certified, anybody looking to make ecological improvements in their land. My pronouns are she, her, and I'm going to pass it over to Nate.
Hi, everybody. I'm Nate. I know many of you, or at least have met you in some scenario or event. My name is Nate Stacy. I'm the Small Farms Extension Specialist for OSU Extension, and my position is statewide. And for the Soil Health Network, I am here just to act in support of all the great work that all of you all are doing. And to Shannon's point, I think we are very interested in hearing what you all are seeing or how you're thinking about implementing soil health practices, what your farmers are doing, what challenges you're having. So to reiterate what Shannon said, this is just hopefully a very interesting and stimulating conversation between all of us to figure out how we can move this science to a certain degree forward, but also the implementation and adoption. I think that covers it.
Yeah.
All to
I
you,
think we're ready
Shannon.
to dive in.
Yeah.
Yeah. Okay. This first section, I know most of you have heard a lot of this. Maybe there'll be some ideas in here that are new, and we're all at different places in our soil health journey and understanding, and so we're going to make sure everybody has that fundamental foundation. It's a little redundant, but we're going to start with just principles and practices. And I would expect most of you have seen something like this figure. This is that NRCS, those basic principles of soil health. And the thing I was planning to say here is what Sage brought up in the beginning. None of these things are new. These are things that we as humans have centered our culture around. Our agriculture has centered around these same principles. I have a favorite book that's called 40 Centuries of Farming, and it goes into like really long, beautiful history. And these are the things we've known. These are what we know is how we can properly take care of land. And there's a lot of different ways to gather that knowledge. And I am very happy that our biological understanding of science is finally catching up to the point where we can say, oh, here's why that works for agriculture. You guys know me. I'm going to start a soil health presentation with a definition, just to make sure we're all there, because I really do like this definition. Soil health is defined as the continued capacity of the soil to function. I'm going to key in on that word function a lot of times, as a vital living ecosystem that sustains life. Without that biology in there, we don't have life as we know it. This is fundamental to everything that we do. As organisms throughout the world. So just so we have that clear understanding, and then we're going to dive into principles and practices. So you saw the slide with the NRCS, 4 major practices: maximize living roots, keep the soil covered, minimize disturbance, support or maximize biodiversity. I added one more in there, that's learn your context. A lot of people who are talking about soil health have included this in these main principles, and I put it here today because we are going to talk about this. It's fundamental for how we're going to be able to do assessments and comparisons and how we're building the database. So I wanted to set the stage here a little bit with context as well. So we're going to dive in, start with maximizing living roots. And for each of these, we're going to kind of do a why do we do this, and then how do we do it? So starting by, why do we do this? Soils want to be covered, right? They want to have a plant covering their surface. Plants are what provide a lot of the foundational components that help the soil to function. So they are there protecting that surface, which is going to decrease erosion, temperature, and moisture. And having that above ground biomass means that we also have that below ground biomass. The roots themselves do a huge amount of the work. Not only are they creating flow pathways that allow that water infiltration and movement, but also create spaces for air exchange and nutrient exchange and microbial habitat. They are physically moving soil particles together and creating aggregates and these homes for the microbes. And one of the biggest things that we talk a lot about in soil health is that these roots are exuding carbon into the soil. And it varies based on plants and all sorts of other things, but about 50% of the total amount of carbon that is fixed by the above ground photosynthetic tissues in the plant are sent down through the roots and even out of the roots. That 50% of that carbon is sent out of the roots. There's no way that a plant would use all of that energy to fix carbon and send it outside of their roots unless they were getting something back. Those plants rely on the carbon that they're fixing from that atmosphere and sending down through their roots to feed the microorganisms because they work in combination. So as we talk about soil health and we talk about getting the biology to do the work for us, a huge part of that is we have to be able to feed those microorganisms. So we need those plant roots to be able to feed those microorganisms. The deeper they are, the more variable they are, the more we're getting different kinds of carbon into different parts of the soil and enhancing that overall ability of the soil to do work because we have fed that microbial community as well as all the physical things that they're doing. So how do we do this? These are the practices that we're all looking for, right? So we want to grow crops in the off season, right? Avoid fallow and decrease your recropping interval, increase time in perennials, manage rotations and forage heights, and really to encourage various kinds of roots so that we have different root architectures occupying different spaces and different root chemistries, feeding different things and creating different symbiotic associations with those organisms. This is commonly done through cover crop blends, crop rotations and intercroppings, any kind of perennial ground cover you can get, hay, pasture, perennials in the rotation. And importantly, especially when we're talking about grazing, when we want to maximize living roots, we want to make sure that we allow those roots time to recover after a grazing interval. So we'll talk about some grazing interval. We want to have short duration, high intensity is fine, but as long as it's short duration where they don't have a chance to go back there and eat that. Often we'll talk about 50% of the biomass. Leaving 50% of that biomass above ground allows the roots to just immediately start working again, that above ground biomass, grabbing that carbon and pumping it down into the ground. And that really has a huge impact on maximizing roots, as you see in this image of what the roots look like after different grazing intervals. All right, disturbance. Why do we minimize disturbance? Again, we're going to talk a lot about microbes, right? We want these microbes to be doing the work for us. And one of the things that they have to do is they build aggregates. They exude things out of their body that glue these different mineral particles together. And that's how they build their habitat. And almost all of the work that the microbes do, it's while they're living in this habitat of their own exudates, these biofilms or extracellular polymeric substance. These things that microbes exude hold that structure together. And that structure is where they are doing the work of cycling the nutrients and communicating stress. And when we go in there and we till the soil up, they're going to be that you're breaking up their home basically. And so we try to minimize that whenever possible so that we can allow those microbes to continue their work. How do we do it? Most of the time we talk about tillage, right? We talk about reduced tillage or a no-till system. Getting a perennial in there is one of the best ways to minimize disturbance. And maybe that's, there's actually no disturbance for 3 years, in a rotation or longer in certain pasture management situations. Sometimes we forget to talk about the disturbance that can be caused by any kind of chemical additions, whether that's an herbicide, pesticide, or a synthetic fertilizer. Those things all have an influence on disturbing the way the microbial community is working in the soil. Another thing here, especially as we talk about grazing, is that to minimize disturbance, we want to consider moisture. And the plant status for grazing intervals. So when we put animals out on the pasture, we don't want to, can you guys see my mouse? If I'm pointing with my mouse? Okay.
Yes, yep. Yep.
We want to minimize these pugging holes, right? When the soil is really soft, the animals are going to create holes in the soil and move things around and smush them. And so, part of the goal here is using plants to help the soil do the function so that we don't have to physically do it for them.
Are there practices that are more relevant to y'all's region or things that come to mind when you hear maximize living roots or minimize disturbance? Yeah.
Shannon, I wonder if you could talk a little bit more about the image we're seeing in the roots and the grass plots there. I mean, I heard what you said and I'm familiar with it, but so it's just the smaller roots that aren't regenerating, or can you tell more about what we're seeing there?
You're talking about this lower image, is that right?
Yes, ma'am. Mm-hmm.
Yeah, so this is from a grazing trial. So we have one area that had, the first one is a plant with the roots along with it where it was never grazed. The next one over is where, it was relatively heavily grazed, but there was a long recovery period after this. And this other one was grazed for a longer period of time and the animals were allowed to come back into it. So typically the growth point on the grass is going to start regrowing at about 3 days. And so if you have animals in a particular paddock for more than 3 days, they're going to go back to that really, really fresh regrowth because there's a ton of carbohydrates in there. It's like candy for them. And so right as that plant starts putting its energy into regrowing that above ground biomass, it gets eaten again. And as we eat more of that, we decrease the photosynthetic capacity. As we decrease the photosynthetic capacity, the plant stops putting its energy into its roots and is really just trying to grow the above ground stuff again. And so there's a reallocation of resources. And as that allocation moves up to the above ground, and there's barely any above ground there, it's hard to get those roots to continue growing. And so when we see that eating again, it just decreases the carbon sent into the roots and through the soil. There are more dramatic images of this, and there's also some really nice time-lapse videos where they show the regrowth of the plants and just the regrowth rate of the plants based on different plant removal ideas. And happy to dig more into grazing systems if that's something that people are interested in.
And Penny, you noted in the chat that going back to new growth is really influenced by paddock size and stalking density. We usually
Excellent.
discuss this in terms of grazing height instead. Is there anything you want to expand upon there?
Yeah, just the distinction between that 3 days mark versus grazing height. It's going to depend on the animals that are grazing as well, because sheep and cows graze differently. So when talking to land managers, we usually talk about grazing height because that's something that is maybe more meaningful for them to measure.
More meaningful than the duration that they're in a paddock?
Yeah, because if I have 5 goats in a really large paddock for more than 3 days, it's maybe not the same as having them in a smaller paddock for 3 days.
Oh, absolutely. I mean, the paddock size is going to have a huge impact. And we can talk more about like that kind of like high intensity situation and how we manage those rotations. Their grazing height is absolutely fundamental. Even if they're in a large paddock, they will find those really sugary spots though again. And so that's part of the reason that we encourage like, okay, you've only got 3 goats. They don't have to be in as big of a paddock and still allowing for that movement because, you know, one of the other things with that is the prevalence of weeds. If they're in that smaller paddock, they're going to eat a lot more of the weeds. And if they're in this big open space, they're just going to find their preferred forbs and the other things are going to continue to grow higher. And so then you have kind of a shift in the species and the weed pressure can be pretty challenging too.
All right, we got Rowan and then the team at Harney.
Thanks, Shannon. I'm curious about on your last slide that we saw, you had consider moisture, and I would think you were referring to that mostly in terms of like a grazing program and possibly compaction caused by the animals or slower regrowth when conditions are dry. One of the things I'm wondering about is moisture as it relates to like microbe viability. And maybe I'm jumping the gun a little bit here, but one of the things that we've struggled with is just, you know, trying to maintain soil health over the drier periods in the summer, you know, with just limited irrigation potential on some of these cover cropped fields. And I'm curious if, you know, what happens to microbes during dry spells and how do we continue to support their health and proliferation if we have limited irrigation?
I love this question. There's a really, really interesting active research on this. And I'm going to take some notes because there are things that we will cover and I'll get more into microbes. But I'm also going to write a note to send you all some publications as I think of them when you ask the questions. There's a really great one called Alive and Kicking. And it's talking about basically what microbes do to survive those hot, dry seasons. And their dormancy and the importance of dormancy in the microbial life cycle. And I'm gonna leave it at that for right now. I did a whole bunch of work and I've got a bunch of slides because of the research that I did at the end. They're not formatted or beautiful, but I've got a bunch of stuff on soil water repellency and what microbes do. It's not just water repellency, but how microbes protect themselves from desiccation and lysis. So we can talk about that as we go through.
Cool, thank you.
Yeah.
I might jump in here just real quick to Shannon, because I think some of the soil health presentations that I've given, all of us have given or participated in or listened to, I think sometimes we forget that with that diagram that has all of those practices, that that's an idealized situation when you can implement all of that stuff and you have time and you have labor and you have money. And soil health is about, like, in my head, and you can all have different perspectives, is about making these incremental changes to your system or to the or providing information for the growers to make these small implementations in soil health. And so it doesn't have to be like, I'm going to cover crop everything now. But I think part of it is like making that first step in these scenarios to try and do something. It may not be perfect, but maybe you reduce your grazing by a day or two or whatever it is, but making those first steps towards trying to improve the soil health. And it's, the change may take time, it may be incremental, but if you're kind of working these different pieces, then I think overall it's a good approach. Not to start to interrupt, but I think we talk about that and we're like, here, do it all. But in reality, for the grower, these are changes to their system. So we have to figure out ways to do that economically.
Absolutely.
Too.
Yeah, I totally agree. We'd often talk about the tuition cost as well, right? Like, it takes time to learn how to do this stuff. It's not necessarily going to be an immediate economic return. You do kind of want to start with a certain set and figure out how to do it on some of your land. And like you're saying, yeah, maybe 3 days is not going to work for your lifestyle as far as a grazing rotation. That's okay. Like, I often will be like giving producers permission for, like, I'm not doing exactly what I need. And like, no, you're making progress. Like, we are moving in the right direction, and let's also be able to take some measurements so that we can demonstrate that you're moving in the right direction. And so we'll talk a little bit more about that.
So my quick question was about native hay meadows and maybe alfalfa as well. So when you go, it's not grazing, but it's, removing the vegetation. So maybe you'll get to it, but how do you manage that when people need to harvest their hay annually?
You're talking about the level of nutrient removals when you're harvesting a perennial crop? Is that right?
Well, I would think that grazing and haying are essentially the same to a plant. In some ways.
Yeah, they're similar. They're similar. It depends on how much above ground biomass you leave, right? As far as how similar it is and what that regrowth interval looks like. But haying and pasture systems can be absolutely beautiful because they do have all of those soil health principles. And so we can let's dig a little bit more into that as we go through.
Yeah, that's fine.
Okay.
I was just wanted to know if there's a difference between haying and grazing.
There's a small difference, and it mostly depends on what your grazing looks like, right? If you go in there and you have everything eaten all the way down to the same level that you would mow to, then there's a lot of similarities. But there is evidence that the animals walking on there and just the impact of the bite actually does have some stimulatory effect for the plants as well. And you're also adding the manure back to it, right? So when you have animals that are actually out there on the land, yeah, they're taking in biomass, but they're pooping a lot of it back out, and they're pooping a lot of microbes on there, and that then creates habitat for other organisms like dung beetles and flies, like things that are going to enhance that overall biological diversity. And so there's similarities in a lot of ways, but from a soil health perspective, you'll often see faster improvement in a grazed system than you would in a perennial hay system. Okay, let's get it back into these here. So how do we minimize disturbance? Okay, that was what we just covered. Now we're on to why do we maximize cover? A huge part of it is erosion, right? If you're a soil nerd, you can think about what happens when a water drop hits that bare soil surface and it dislodges mineral particles going this way and that. And, that leads to erosion. It is part of that disturbance effect. And so we want to be able to minimize erosion whenever possible from wind and from water, which also helps us to reduce inputs because they're staying where they're supposed to. There's a huge difference in the rate of water infiltration moving into the soil when there is a cover over the top of it, whether that's a mulch or a residue or a compost. It's very different. It can also decrease that crusting that you often see. That's another fun topic. It provides habitat and food for soil organisms, right? We want to have a diverse set of organisms that are going to be able to do each of those functions in the soil, right? And that's going to help for nutrient cycling as well as decreased chemical dependency, which kind of goes to the next point as well, where having a cover over that soil surface is going to decrease your weed germination, which can also help us to decrease that chemical dependence. There's also a huge moderating force for both soil temperature and moisture. It creates this hydraulic barrier, basically. The sun's not beating down on a dark mineral surface, and the water moves through soil. We've talked about, capillary action. We understand that soil is going to move, water is going to move through that soil and evaporate. Well, if it's moving through the soil and then it hits this really, we'll call it fluffy or non-contiguous type pore structure, the water doesn't have that same kind of ability to move out of the system. So it can really influence moisture status for your soil. How do we maximize cover? Again, pasture, hay, perennials, that's the way that you can have plants growing all year long. And I would say plants are the best way to maximize cover, but they're not the only way. We use plants. We have the perennials, the cover crops, intercropping, conservation cover, but residue management is also a way of covering. Mulching is a way of covering. Even, a black plastic mat can be a way of covering and protecting that mineral surface. Talking about livestock, we do want to think about controlled traffic. So up here in this picture with the jerseys, we're moving livestock on a regular basis. It helps to have controlled traffic, have some pathways, have some areas where they're moving, and that kind of goes along with those sacrifice areas. If you live in Western Oregon, there's going to be some parts of the year where you don't want to have animals out there because they are going to be doing damage to the soil. And so rather we might have a small zone where they are for a really wet period so that they're not making bare spots in the soil. That's going to help with the cover. And then the timing of grazing as well has a huge influence on that regrowth potential and the soil's ability to stay covered in a perennial system. Why do we maximize biodiversity? I think there's a lot of people who say, I don't really quite get why I need to have all of these different things in here. As we talk about soils and we talk about function, one of the things that I like to bring up is that only about 1% of soil microbes have been described or can be, in pure culture because they need each other. They're like, an individual soil microbe has the ability to do maybe 1 or 2 enzymes or, acids that they produce. They don't do a whole lot. There's not a huge amount of genes in there. And so they're almost like the individual, like the members of a biofilm in a soil microbial community are almost like the individual organelles in any kind of mammalian cell, right? Like there are some of them where this is where the information is stored. Some of them where they're in charge of waste disposal. Some of them where their job is to attach and ground and build structures. And so we can think about it either as like the scale of an individual cell, or like if you were making a Sims city and you wanted to make sure that all of the different things that the community needed were taken care of. That's why we need diversity in the soil, because each of these organisms is doing a different thing. And in order for the community to really thrive and survive, we need, it takes all, right? Like it takes all kinds. And so that goes towards, all the different kinds of organisms that are part of that organic matter recycling. It also really is a strong thing for chemical communication of stressors. Maybe there's some soil nerds on here who want to hear more about quorum sensing. Quorum sensing, when the microbial community gets to a certain level of community diversity and evenness, they are communicating with each other with chemical signals to let them know about stresses. And that doesn't happen when the community is really skewed one way or another. And that has a huge influence on the soil's ability to to resist disease or soil suppressiveness, disease suppressiveness, as well as pest suppressiveness. And part of that communication then goes into like the plant stress. When the plant is stressed, it sends out signals and the insects come and the diseases come. And if we've got that microbial community doing the things that the plant needs in order to survive, and the plant doing the things in order for the microbes to survive, we don't have that same level of stress, and we often see a lower disease and pest influence. So then how do we maximize it? We want to grow diverse cover crop blends or diverse pasture rotations, right? And as we do that, we want to think about different kinds of functional groups. So thinking about functional groups, grasses, legumes, forbs, brassicas sometimes are in their own group separate from forbs. They each do different things in the soil and they attract different kinds of organisms. They have different kinds of exudates, and that has a huge influence on what that diversity looks like underground. We can do this through rotations, through livestock grazing, through cover crop blends, increasing the time of perennials in any kind of rotation. And we want to really think about making feed for our target organisms. I come from a beef cattle background, so everybody knows the importance of the feed that we give our livestock in that world, right? But I like to think about feeding that microbial herd as well. What are we feeding the microbial herd so that the soil is doing the work for us, so that we can have a decrease in inputs? And part of that, we're feeding them and we're protecting those structures. So from there, I would just say that there are some practices that are kind of common to all of these principles. Anybody want to jump? Oh, no, it jumped in first. Just plant more plants, right? This is kind of like all of it. Just keep your soil covered with live plants as much as you can. But obviously it's not that simple, right? It's never that simple when we're actually doing it. And it takes intention, and it takes like figuring out which plants are going to work in which ecosystem and which ones are serving the purposes that we're looking for. And so I think, maybe we'll have a follow-up later after this, nicely laid out like series where we just talk about plant selection and what plants are going to work in which area for which systems. And that can be another 2-hour session. Questions on basic principles and practices. This picture right here, these are both from the same farm. They are both wheat production systems. This one's just a wheat fallow system, and this is a wheat with a cover crop system. And I love that side by side of these two fields literally right next to each other.
I always think too that like sometimes soil health, if you go into the practices and all the things you're supposed to do, it's sometimes it's overcomplicated in a sense. Like you just have to grow plants. And the other flip side of the coin is that the pictures that Shannon just put up is that right side of that screen, that's what we're trying to avoid. Even if you don't know what poxy is or respiration is, you can look at that and be like, this is probably not what my soil should look like. And sometimes it's easy enough just to think of avoiding that and instead of like, I need to do this and this and this, when really we're just trying to avoid that picture right there. And in any way that we can, and any step towards that is a good thing.
So I may need an offline discussion on that, but the type of cropping that I work with most, it's maximum disturbance, maximum, a lot of stuff. It's against the principles because they're growing seed crops. And a lot of these are annual plants, so they need clean fields, and they can't bring livestock in there who could poop out weed seeds and all that other kind of stuff. So my big challenge is trying to help with soil health in a agriculture that is that a lot of these practices are not going to be used. So I don't know if that's unique to us in Central Oregon. I would guess there's got to be other seed growing areas in the valley where you have a lot of these issues. I don't know. So I don't know if my big issue is something that is relevant to others in the group, or if I need to individually work with you outside.
Ellen, I would 100% agree with what you're saying. It's really hard to use soil health principles in seed crop production. It's not to say that it can't be done. For the last 3 years, I worked at a seed company. And so we talked a lot about this. I talk with our production, our field rep basically, and talk about how we can do this. And I think you're right. There's a lot of seed crop. Like Oregon's one of the biggest seed crop producers anywhere around, whether it's, over in the Blue Mountains or Jefferson or the Willamette Valley. We have a ton of seed production. And so when it comes to unique challenges, I think it's one that we should dig into. And I'm putting that on my list of things to follow up on.
Thank you. That would be so helpful.
Well, yeah, that's really helpful feedback. Also, just to let you know, I'm relatively new in this role. And so in extension, the first thing that we do is what's called a needs assessment. Anytime you all have questions or thoughts on what you need from a soil health extension specialist, please do not hesitate to tell me. And if it is a special thing, like I can go to the seed growers and say, hey, the folks who are trying to do this work need this kind of information about soil health in seed production, and let's figure out a way to get some projects going on it.
I think the benefit too of like having, my opinion, having these discussions is that Ellen, like maybe there's a way we figure out some type of research project or some type of intercropping that might work that somebody hasn't tried that we could experiment with. I think that's part of what the entire soil health network is that we're a group of people who are invested in this, and we're going to have to figure these out these challenges if we want to move this forward. And there are going to be hard things. Like that's just the reality of the situation we're in agriculture. And so maybe someone has a great idea. It hasn't come out yet.
So we've talked about the principles and practices. This is probably something you talk about with your producers on a regular basis. I have spent, what is it now? The last 12 years really working on soil health assessments. And so, the first question is, well, why do I really need to do a soil health assessment? I'm doing these practices, isn't that good? The first thing that people want to know when they're talking with me about soil health are these actions that I'm taking actually making my soil healthier? Is this practice going to let me decrease my fertilizer applications? Is this practice going to allow me to have more disease suppressive soil? Is it going to be more resilient when we have a bad drought year? They want to know if we're getting towards healthier soil. When I think about, is it actually moving towards healthier soil? I want to think about, is this management improving my soil's capacity to function, right? Is this moving my soil to having a more, vibrant, living, vital community? And so that's where we kind of come to the point where if we are going to do this soil health assessment, the reason the producers want to know is there are always trade-offs and benefits when it comes to, okay, we're going to take on this practice, and that's going to be hard for X, Y, and Z reason. In order to justify to your business partner or your dad or whoever else that you should be making this change, you need to be able to demonstrate that it's going to help you in the long run, because it's going to be hard in the short run. And there's going to be things that you have to learn, and there's going to be things that maybe look terrible for a year or two. And in order to have the confidence to keep moving forward along that journey, it really helps to have some quantification and a goal of where you're going. Okay. There are other reasons that people want to do it. Maybe the major goal is for marketing. There are people where a huge part of the marketing is their customers are asking for a certain kind of stewardship. If you've been doing this work in your land for a long time, it's great to be able to show that result or to show the progress that you're planning to take. And then, this last point kind of comes to this quote from Maya Angelou: to know where you're going, you have to know where you've been, right? In order to understand our context, we have to understand what where we have been and where we're going to go. But why does the context matter? And I think I'm talking about just soil nerds. You all know soils have a huge amount of variability. With soil health assessments, we almost always have some kind of comparison. Maybe it's over time, maybe it's the same field, before and after certain management practices happened, or maybe it's two fields on your property that you're managing differently. There's almost always this comparative element within a soil health measurement. And soils, because of this huge amount of variability, and they have an inherent capacity to function differently. This really shallow soil over here in Jefferson County in Madras has a different amount of area, just straight up area that the roots are going to be able to explore for nutrients and water. It's going to be very different. This soil here in Philomath, there's going to be challenges with getting through these rocks. We have differences in the water holding potential just within this Willamette silt loam, where we have layers of much sandier soil that are going to dry out faster. Huge amounts of carbon here in the top, right? Like each of these has very different capacity to function. And as we think, the reason we need to think about the context is because our goal is to say, okay, within my site, within the soil type that I have and the climate that I have, is there a difference? And so we have to be able to pare it down into some kind of soil health sampling groups so that we can characterize changes in an individual soil's ability to function. That's that dynamic capacity to function. When I talk about inherent, and I'll probably use this word a lot, inherent is like the soil's natural inclination. It's like there's a certain range that every soil can have, and they're not ever, a sandy soil might not ever be below 0.2% soil carbon, but it might not also ever be above 5% organic carbon. Where if you have a high clay soil, you might be at a 0.5% for a minimum soil carbon, and you might go up to 20 for your organic carbon in that clay. So we need to know what those limits are.
It's like having different potentials, right? Would you say, like, if you were trying to think about it another way, like this soil has this type of potential, and other soils might be constrained just because of physical differences or chemical differences, and it ends up all being related anyway.
Yeah. And when we look at like the data on this, if you just like lump everything together, the inherent properties are much larger driving factor for what you see in the soil health analyses than the individual management practices. And so in order to see the differences by management practice, we have to have the soils grouped. Texture for a long time.
Can you say that again? Because I have to remember that. Like that point I think is critical, that there can be a limit to soil health by a texture class or whatever it might be, that you can do everything right, and whatever value you think you should be getting to, it may not be possible.
Yeah, absolutely. We kind of want to put these bookends on what the potential is for each soil. And so that's kind of where we're going with a lot of this database and the context, and we're going to get right there. This texture has been this primary grouping for, especially in the Cornell assessment of soil health. The first thing is just fine, medium, and coarse textured soils. Texture has a huge influence on the soil's ability to do all sorts of things, whether it's available water holding capacity or carbon storage capacity or nutrient cycling rates. Texture is a huge driver. As we're thinking about soil health samples, we're normally either in the top 6 inches or the top 12 inches. That's where a huge amount of that biological activity is. And so rather than grouping by soil order or grouping by soil, like any of the other classification series, texture is one of those where it has a huge influence on that biological community, habitat structure, and then the soil's ability to function. In these two figures here, these are both from the North American project to evaluate soil health measurements. I'm going to call it NAPSHM from now on. It is the big project that I worked on with Soil Health Institute. There's a lot of data in this project where we literally went to 124 long-term agricultural research sites across the country, sampled 2,000 samples, 2,000 experimental units in 1 year, and then ran about 150 different analyses on all of them. So there's a huge amount of information on the measurements. The reason I bring these two in right now, we have intact available water holding capacity. And so we have, this density distribution of the available water holding capacity for all of these soils that have a coarse texture, so more sandy, or a medium texture, or a fine texture. This is that difference in potential, as well as difference in kind of propensity to be there. We often talk about soil carbon when we're talking about soil health. This SOC, soil carbon. Soil carbon and total nitrogen have a very close relationship, as I'll show you in a couple other slides. And a fine textured soil, yeah, it can be very, very low soil carbon and soil nitrogen, but it also has the ability to be very high soil carbon and soil nitrogen. And of course, textured soil is just never going to get there. Soils have a different capacity to store carbon. We often talk about carbon as like this thing that we need for soil health, but not all soils can get to the same level of soil carbon. And so there is kind of an inherent capacity or potential for carbon storage. And this could also look like a precipitation map of Oregon, right? It could look like a net primary productivity of Oregon, right? Like there are a lot of different factors that are going to influence the soil's organic matter concentrations. When we get to a small scale, we can see the differences in the dynamic management related properties, but on a large scale, there are just some things that are ruled by climate and soil edaphic characteristics. This is one of my
Shannon,
favorites.
can you go back to that
Yeah.
slide? Is this based on Marcus's paper?
No, this
No?
is
Okay.
this is NRCS data that is on the California Soil Resource. Rather than using the Web Soil Survey map from NRCS, I use the California Soil Resource, but it pulls all of the data straight from the Surgo database. And so you can go into this website, you can do it by different depths in whatever kind of area you want. You can zoom in as much as you want. But this is mapped data, not Marcus's data.
Great. And can you explain a little bit why yeah, I guess you kind of already did touch on this, but
Yeah,
why
I mean,
certain parts of the state have different capacity to store carbon than others? Is it
Yeah.
the type of soil and the texture?
Yeah, and I would say moisture is one of the biggest things, right? In order for a microbe to take organic matter and use it and blow off CO2, sorry, I'm gonna say it a little differently. Every organism has a need and is related to the moisture and temperature. When we're talking about who's going to function where, moisture and temperature are always going to be influential, whether we're talking about bacteria or fungi or an insect. Moisture and temperature have an influence on their metabolic rate. And when we're talking about metabolic rate with soil microbes, often we're talking about taking carbon in and blowing off CO2. If the soil is saturated for a good portion of the year, there's not oxygen. And so that metabolic rate really slows down. And so when we have areas with high rainfall, or we have areas with impenetrable surfaces or subsurfaces where the water is just standing, then we have a real decrease in the aerobic or oxygenated pores in the soil. So we have a decrease in the rate that carbon leaves the system. Often having that higher precipitation also has an influence on higher primary productivity, right? We need rain or we need moisture in order to grow these plants. And so when we have more moisture, we have more plants, we have more roots that are exuding carbon into the soil. So they're putting more carbon in. And if they're in that period where there's a lot of what we call anaerobic microcytes or small pores that are fully saturated with water, they're not going to be losing that carbon as quickly. So it's a balance of both the amount of carbon going in and the amount of carbon going out that is highly related to the amount of water that's in the soil profile at any given time, which then is also related to texture.
And this relates to how you need plant roots to feed the microbes, and the microbes bring carbon into the soil.
Yep. And yes,
Mhm.
absolutely.
Mhm.
So this is for all the soil nerds out here. Ready? So, oh, just a second. This didn't give me, shoot. Okay, I'm just really frustrated because there's supposed to be 2 slides on here and it looks like, or 2 images on here. I'm gonna introduce this while I am taking just a tiny, oh, sorry, just a second. Just a second, I just have to find this other image because it really makes this a lot better. Shoot, I'm probably still sharing that screen. Okay. I am going to not worry about it. I'm gonna just, oh, there we go. can you guys see that right now? That's the
Yeah,
image that I want.
Yeah, I see two. Oh, I see what
Okay.
you're saying.
There,
Yep.
there's different colors on these. Okay, so this is a map of North America is colored by the Hargreaves climate moisture deficit. So sites that are dark blue are very wet, and sites that are red are very dry with a gradient in between. The black dots are each of those 124 different long-term agricultural research sites. You'll notice there's only 2 in Oregon, so we need to do more. But it represents North America. What all of these dots are over here, this is what's called a detrended correspondence analysis. And you don't need to know what's going on with a detrended correspondence analysis, except for that the closer 2 dots are to each other, the more similar that microbial community is. Each of these dots represents an entire bacterial community from one sample. So there's about 2,000 dots on here. This is the entire bacterial community for this one, and this is the entire bacterial community for the other. So these dots are also colored with the Hargreaves climate moisture deficit. And the reason that I show you this is because the climate, just the climate itself, has a huge influence on what that microbial community composition looks like. So the community structure up here, these guys are very similar. They're in a very similar climate, and their microbial community is similar. Over here, these guys are very similar climate, very similar microbial community. You do see some areas where there's a difference in climate, and they're close to each other, right? So there's some interesting stuff to dig into on those. And you'll see, some yellow here and some yellow here. So there's some interesting. The other factor, so I have climate and pH. This is the one where we see a really, really strong gradient in what's going on in the microbial community. So now this scale, we have a different scale, same exact sites, same exact samples. The scale over here, this is really high pH and this is really low pH. And you see how influential pH is on driving what that microbial community structure looks like. And so I say this too because we talk about, cycling rates or moisture or carbon, but the climate and the pH themselves are influencing the community structure and who is doing what in a soil environment. And it brings us to this next point where it's like, okay, we need to be able to group soils because over here on this big scale, on the grand scale, it's climate and pH that are driving it. But we want to know about those dynamic characteristics, right? And so once we get into a single site, then we can do the same thing. This is also a detrended correspondence analysis with the bacterial community. But now we see this differentiation by treatments. So we have 4 different fertilizer treatments here from a field in Idaho where there's lots of dry dairy lots over there, and there's a lot of manure that gets spread in agricultural fields. And so these 4, these are from a part of the field that these are for the 4 treatments that have no fertilizer. These are the ones that get synthetic fertilizer. Here's our 10 ton per acre of manure, and here's our 30 ton. So once we say, okay, these things are driving it on a large scale, we need to be able to group it so that we can then see what's going on with that dynamic characteristics. This brings us to a plan for having these soil health sampling groups. I will tell you, this is a very, very active area of research. The paper that this comes from, it has not been published yet. There's other people in Washington who are using kind of similar methods, but a little bit different science. And there's a lot of what's the best way to do this? And there's a lot of different ways to analyze it. And so there's a lot of different answers on what is going to be the best. There are multiple systems. Nate and I are both part of groups who are trying to make sure that we have as much consistency within how we set up these sampling groups, so we have as much cross comparability as possible. This is a map that is created by the Soil Health Institute with the soil health sampling groups that they have determined. And this is, you can use the same algorithm basically to map this anywhere. I just had it done for Oregon. So the main 2 characteristics that we have in here are the soil texture. And instead of grouping it into 3, we have it grouped into 6 different soil textures, and then 4 different drainage classes. We talked about the importance of those anaerobic and aerobic pores in the soil. When you have a really poorly drained soil, you have a increased storage capacity of carbon, but that doesn't mean your soil is healthier, right? And so we need to make sure that we're grouping it that way. And so we go from having about 2,000 different soil series in Oregon to going to 24 potential groups. And then we layer on one additional thing, and that is using MLRAs, or major land resource areas. A clay poorly drained soil in the coast is going to be different than a clay poorly drained soil, outside of Klamath Falls. And so we have these major land resource areas, as well as the texture and drainage. And those are the three characteristics that we're using to create soil health sampling groups that are big enough to have some a minimum data set where we can actually compare and understand what the range and characteristics would look like, as well as being fine enough that we can actually see differences in those dynamic management practices rather than just the inherent properties of the soil.
So Shannon, basically in the last, whatever that is, 5 or 6 slides, that's X number of years of work just to figure out how to group things so that eventually we can say that you should use 3 cover crops or 2 cover crops. And that's not that's not a criticism. That's just the reality of the thing that we're doing. I think that sometimes it gets glazed over too quickly that this is a very, obviously, I think we're all soil nerds here. It's really complicated, but to be able to make or to have guidance or to have suggestions for a grower to make an improvement, you still have to understand the basics, and you have to be able to account for the basics. And so it's like I work like tangentially to this. That's, years of research just to figure out how to group things so that we can make a reasonable suggestion.
Right. Absolutely. And I would say, we have these principles. We've got principles that, we've known throughout history, and we can tell people that this is what is good. But I often think that there are a lot of people who are data driven, and in order to say, okay, yeah, I'm ready to do that hard work that might take me more time or more energy, or, hit my yield for a year or two, then we have to be able to show them the data in terms of these outcomes that they really care about. And you're not going to see any differences in outcomes until you group it, right? And so, this is very active. This is work that I started when I was at SHI in 2021 on this paper that's just about to come out, right? And before that, I had a whole different scheme that I was trying out with Oregon Soil Health using land capability class, right? It is an iterative process. We're trying to figure this stuff out as much as possible. And I don't think we're all the way there yet, right? I know that there is still ongoing work around this. One of the things I'd really like to see is for all of these different soil health research groups to come together. And I think that's why we have this power in the soil health network that OrCAN is helping to coordinate here, is that at least in Oregon, we can have a similar set of doing things, right? That's part of that goal with this collaborative database, is that, okay, maybe we can't do what everybody does all throughout the entire country, but in Oregon, we can have a database that is going to have intercomparability no matter who's doing the work in the sampling. So once we have them grouped, then we can be able to show a producer something like this. And this is, we're going to talk about indicators after the break. This is the maybe last slide before the break. One more slide after this. And what we have here, we have 2 soils. These are both from CBARC, the Klamath Basin Agricultural Research Center in Pendleton. This has been in a wheat fallow system over here for 80 years. And this has been in a perennial grassland, mixed species with a tiny bit of grazing in it for the last almost 80 years. So from this, we can kind of see those bookends. We can kind of see what the potential of this soil is. This is just on one farm. And you can see the difference, right? If as a soil health practitioner, you're like, oh yes, I can tell that one of these soils is healthier than the other. But it gives us these limits. It gives us this upper potential and lower potential of what is possible for this soil. We have very long-term consistent management where we have done no soil health practices over here for the last 80 years, and basically adoption of all the soil health practices over here for the last 80 years. And so we get these bookends. We say, okay, for the total organic carbon for this Walla Walla silt loam in Pendleton area, we're probably not going to get much below 0.8. We're also not going to get much above 2.2. So if you're at 1.7, you've made really good progress, right? And this sample data is also from the exact same farm. This is in a wheat pea rotation with no till. So we go from a wheat fallow with chemistry and no residual inputs and all the synthetic fertilizers and chem to a wheat pea rotation with no till. And then over here to a mixed species grassland with very occasional grazing and typically just mowing about once a year. We see that for organic carbon, respiration, and aggregate stability. The sample, they're all in between what the bookend potentials of the system are and show us that like progress of what we can get to. These indicators are great, but what the farmers care about are the outcomes. And so we're going to be talking more about outcomes. We're going to talk more about outcomes, and what we want to say is, okay, how does this actually, what does this actually look like when it comes to something like available water holding capacity or saturated hydraulic connectivity, and how can we measure both that low end and upper end, and then know where the soil sample is for the producers in the measurement that they need in order for their bottom line to really matter. Now we're going to have any questions on inherent capacity, soil health sampling groups, and have the break. This is carbon stocks for the whole country. Just so you know, like this map right here, and this is again from Sturgo data. You can notice that when you look at just Oregon, we've got all the opportunities, right? Like we've got all the variation within our state. There are some states where it's kind of all just similar capacity. Oregon's huge amount of variability makes this like one of the most amazing soil health testing grounds and ways that we can take what we do in Oregon and apply it all over the country.
See, Rowan has your hand up, and Rihanna, I saw you unmuted if you want to go next.
I'm curious, Shannon, what sampling methods you're using to get the CO2 respiration and the aggregate stability. Is that PLFA or something else?
Nope, we're going to talk about indicators in the next section. So I'm going to punt that for now, and then I do also have like a whole thing on sampling protocols. So we can talk about that maybe at another one. I did not put sampling protocols in this presentation. I would love to, Sage, I would love to have a session where we meet in person and we do training on sampling protocols so that we're all on the same page and doing things in a similar way. So again, yeah, the methods matter when we're doing these things, and I want to have that comparability.
Cool, thanks.
Yeah.
Rihanna.
This is so great, Shannon. We've been so excited to learn from you. So thank you for all your sharing. Would you mind going back to your previous slide? I got a little confused about what the treatments were on the what I'm understanding is that on our left with the mud and the shoe is the growing wheat and then fallow, and on the right was the mixed, more perennial natural system, or I got confused about where the peas
Yeah,
came
sorry
in
about
on
that.
which one of these. Do you mind?
So I
telling-
don't have a picture. I don't have a picture here of the sample field of interest. Okay, but the production for this is a wheat pea rotation, so there's no fallow in it, and it's in no-till production. So it's in a no-till system with a wheat pea, and that is our soil health sample of interest. And then
Oh, I
this
see.
low.
So the sample is not either one of these pictures. The sample is actually a third
Yes.
management. Oh, okay.
Yes. Yeah, the sample
got
is a
lost
third
in there.
management. I just didn't get it nicely onto this image. I will grab another slide that I have that I think you'll like because it might make it a little bit more clear. But over here, this is that low end wheat fallow, and it's a tilled fallow, but there's also chemical applications. There's also synthetic fertilizer. And this one doesn't really have any inputs. It really just gets mowed about once a year for fire mitigation challenges. Back in the 80s, it was grazed for a couple of years. But other than that, it has been in a perennial grass mixed species area.
Okay, and that's my question is I just wonder like why for a mowed mixed grass, presumably distressed, uncared for, maybe. I just wonder why that's the measurement for the upper potential when they're not actually exhibiting the principles that you were saying, which would be optimal, with the variable root depth and perennial and the different principles we covered. And so I'm just wondering, why are we using that sample to determine the upper potential of that?
I have one slide I'm gonna grab for you as soon as we're on the break so that you can see it. I would say that a well-managed grass pasture or mixed species pasture has about the highest soil health I ever see in any system. The absolute worst soil health I ever see in any system is also a poorly managed graze system. And so I have the other slide has more production systems in there and kind of a more full range. This is kind of just an idea of these bookends. And frankly, the reason that I don't have this upper potential is because on this farm, on this day when I was saying, like these two pictures were taken on the same day, on the same farm. And so there's a certain comparison there. If we're looking at the overall upper potential, and we can talk about this when we get into database stuff, I would like to see it as a little bit of a different system. But a perennial grassland does take on most of them. Like it's covered year round, it's got roots year round. It's serving a lot of the principles, but it's not quite as high as what we see in some of the grazed systems.
Thank you. That's really helpful. Appreciate that.
Yeah, and I'll grab that other slide.
So often it feels like we're repairing these mostly invasive grasses, pasture leftover systems. And so it's good to hear those opportunities and for habitat and soil health.
Yeah, it really, grazing spans that potential, upper and lower potential. Literally, the worst soil health is in some really badly grazed systems. And there's also no way that any row crop system can ever get to a beautifully managed grazed system. It's like not even possible within their potential. So there's thoughts on that as well. So this is reference-based interpretation. Part of my goal in the soil health database is to be able to have each of these different kinds of management things kind of characterized along this line. And here we do have one of the outcomes, right? Saturated hydraulic conductivity, the rate that water is moving into the soil. That is something that matters for producers. And over here, again, like I said, where is it? Where's my mouse? There we go. We've got that poorly managed system. Then we've got a wheat fallow tilled, and then wheat chem fallow with no till, and then maybe a wheat pea with no till, an organic wheat rotation that is also cover cropped. And then we've got a mixed species, not actual agricultural production. And then we've got well-managed grazing. And so there is that whole spectrum. And so I appreciate you interested in that part because this is the goal. This is where we want to get for each different kind of soil within their soil health sampling group and climate. We want to be able to show somebody what that continuum looks like as they're trying to decide, what they want to take on. All right, so we're going to talk about observations, indicators, measurements, and outcomes. So the first thing I want to say about this is, especially as people who have studied soil science, our observations can tell us a huge amount, right? Like we know what a healthy soil looks like. We've seen it in pictures. We know a lot of these things. And so observations can be a huge part of soil health assessments. The indicators that we talk about with soil health, that's an inference. We're making inference about the soil's ability to function based on some measurement that we think is related. There are certain things that we can directly measure. And when we talk about, we talk about even the CO2 burst test, right? That is indicative of microbial activity. It's not just a straight measurement of activity, right? But we can take an actual measurement of bulk density. We can take an actual measurement of, the moisture content in a soil at any given time. My goal is to be able to connect these management practices and the quick and easy indicators that we have available to the outcomes. So we say, okay, this management is giving us this amount of water infiltration, and here's an indicator we can use so that we don't have to do like an extensive outcome measurement. Doing water quality work and like setting up measurements from tile drainage is expensive and time consuming. We can't do that on all of them. So we want to be able to have an indicator that is well related to measurements of the actual outcomes.
And Shannon, outcomes could be, because I think this part gets somewhat confusing, not confusing, but we've really focused on the indicator part for a long time, but the outcome could be, it could vary, right? By region, somebody could be more concerned about water quality, less concerned about yield. Maybe the outcome could be a number of different things that are particular to even to a grower, I think, right? I mean, they're going to utilize soil health to get to an outcome that they are interested in. Oh, perfect.
Yeah.
Yeah.
So this is one of the things. So when we're talking about outcomes, these are things that I hear from producers on a regular basis. Like, number one, it's going to be economics, right? Like, we have to be able to talk about what the profit and yield, like the influence of soil health management practices on our ability to make money, plain and simple. But for a lot of people, especially right now, we hear about the challenges with drought, right? We need to make sure that we get every drop of water that falls out of the rain, that it infiltrates into the soil, that soil can then increase its water holding capacity. And there's a lot of challenges too with water quality coming out of agricultural fields. So that soil's ability to filter, like these are the outcomes that in the soil health database, we're going to be having measurements of the actual outcomes as well as measurements of the indicators, so we don't have to measure all of the outcomes. We want to be able to make that relationship clearly understood from the indicators. There's kind of this circular logic that has happened in soil health for a while where we say, oh, these are the things that you should do for soil health. These are our soil health practices. And when you do these practices, we have higher POC and we have higher respiration. And so higher respiration must mean we have good soil health. And that circular logic is challenging because we need to know if it's actually increasing the soil's capacity to function. And these outcomes are those soil functions. These are the things we need the soil to do. And so we want to be able to measure the functional outputs of the system. I'm going to just, okay, so we have way too many slides. I tried to make it short, but okay. So I'm going to start here with infield assessments. Anybody who has done soil science knows that there's a huge amount of information that we can get just from doing our own visual and tactile assessments. There are some measurements that we can do out in the field. You can do an aggregate stability test out in the field. Infiltration almost has to be done in the field. Compaction has to be done in the field. And when we do our in-person training and we're doing measurements and sampling plans and how we do these methods, I would love to take everybody through an in-field soil health assessment where we kind of look at a rating for soil cover and ponding, soil color, structure, worms, other organisms, pores and roots and smells and feels. Like all of these things are influential. And to be honest, a well-trained soil science can give you a soil health score from these kind of things as well as any lab measurement can, but that's not nearly as quantifiable. And so when we're, looking towards other stakeholders or, there's somebody who needs it for marketing or just because they are data-driven, they need harder numbers. There is a
Shannon, you
field assessment from NRCS.
Shannon, you mentioned POC. Can you explain what that is? Define that.
I will in just a minute.
Okay,
Yeah.
perfect.
There is an infield assessment worksheet from NRCS. I will say it's okay, but just yes or no on each of these is really hard for a lot of people. And so I have looked at a lot of different infield soil health assessments. This is kind of my working sheet for what we have right now. But we can talk about that more again later when we're doing work together. Soil health assessments, typically it was kind of grouped into like the biological, physical, and chemical, and we needed to have a whole bunch of each of them. And Cornell has done a huge amount of really beautiful work in the system with these 12 major indicators. This is what that Cornell soil health assessment looks like, but I also want to point to the fact that a new paper that is by Cornell using 6,000 samples from the comprehensive assessment of soil health, its major conclusion is that a simplified soil health assessment can be performed at a low cost, possibly in the field, and doesn't need nearly as many measurements. So Cornell does offer these, and they offer kind of a basic package, a standard package that also adds respiration and ACE protein and available water holding capacity, and a plus package. So what we're going to do is we're going to talk about the kind of minimum data set, some of the major indicators that have been shown to be really, that we think are the best indicators of soil function at this point.
Ooh, Shannon, but the Cornell system, the data that they use is really focused on that region, right? So any of their distributions, all of their, not all, but a majority, yeah.
But in this paper, stat scoring functions and regional analysis, they did have 3 regions. So they had a Midwest region, a Northeast region, and a Mid-Atlantic region in there. And there are differences by region, but absolutely. I mean, Cornell doesn't even talk about, you know, calcareous soils, right? Like that's just not even part of it. And we need to have different grouping for calcareous soils. They perform differently, they act differently. Like that's not part of their world. And so absolutely there are their distributions that they have set up are all relative to the samples that they've taken. So it's all comparative. Okay, so just some kind of basics. Soil organic carbon. Roughly 50% of organic matter is carbon, is this organic carbon. And when we're taking a measurement of soil organic carbon, it is that net balance of inputs and outputs of carbon in the soil in a long-term kind of setting. Soil organic carbon is going to be one of your slowest indicators to actually change. It is that concentration of overall carbon in the soil. And so I often think about it as the overall potential feed for that microbial community. That carbon is there. There's a chance that it could be feed. The machine that is typically used to directly measure soil organic carbon combusts all of the carbon. And so if you have calcareous soils, if you're ever at a place where you've got pH over 7.2, you drop some acid on the soil and see if it bubbles. If it bubbles, there's carbonates in there, and you need to be able to subtract the concentration of carbonates out of it in order to get to just the soil organic carbon. In that machine, they often also measure total nitrogen. So it's kind of nice to get the total carbon and nitrogen. Soil carbon is related to basically everything. There's an indication there, but we have to make sure that we are doing it within our sampling groups, because otherwise it is very, very reflective of the inherent soil potential. Considerations, there's a huge gradient in carbon as you move down in the soil profile. So as we're doing comparisons, again, we want to be fair. We want to have the same depth. Typically, we talk about more carbon is better. It's very slow to respond, and there's a lot of inherent things related to soil carbon concentration. This again is from that NAPSHM North American project to evaluate soil health measurements. So 2,000 samples here, where we're taking a lot of these different indicators and looking at the Spearman correlation between them. You see organic carbon is pretty highly related to almost everything. This is this is from a paper that again came from that NAPSHM program. And what we did was we reevaluated what we call pedotransfer functions, a fancy way of saying you use soil properties to try to predict another property, basically. And so we have the predicted available water holding capacity. And in the past, I forgot to put this on here. The red line is the old formula. This is the old pedotransfer function. It did not take into account soil carbon, because in the past, when we measured available water holding capacity, we ground up the soil, packed it into a plate, put it on a pressure plate, and measured how much water it could hold at field capacity and wilting point. What we did in this project instead was we did that field capacity measurement while in intact cores. So when you have carbon, a huge part of it is building that structure and building that aggregate space and allowing those microbial exudates to hold that water. And so when you measure field capacity using an intact core, then all of a sudden the amount of carbon that's in the soil has a huge influence on your available water holding capacity. So in this figure, we have soil organic carbon at 10 milligrams per kilogram, 20, 30, and 40. And you see that increased available water holding capacity for both non-calcareous and calcareous soils. Again, in the Saxon there's no difference in calcareous or non-calcareous. We did find a difference in our data set. And there's a huge difference, especially in the non-calcareous, in available water holding capacity by just by including carbon concentration into that pedotransfer function. CO2. This is basically I get frustrated with this because there's way too many names for it. That's why up here on the top, these are the different names for CO2, right? Most of the time it's been called respiration. That's fine. There's some people who are going to have a problem with that because not all of the CO2 that's coming off is actually directly microbial respiration. Haney also, kind of has this as a central piece in their soil health assessments. It is this, and they use a 24-hour CO2 burst test, basically, where you have a soil that's sieved to 2 millimeters. It is air dried, and then we add water back to it. We add water to about 50% water-filled pore space, and then we close a jar, basically, and we measure how much CO2 accumulates over a 24-hour period of time. It is highly related to microbial activity, microbial biomass, and the rate of nitrogen mineralization, or the rate that we turn soil organic matter into plant-available nitrogen. It is very different. The laboratory CO2 test is very different than what we see in the field. Often when we have a laboratory respiration test that is very high, we'll often see really low CO2 coming off of an actual field. Again, we can talk about that in a lot more detail if anybody's interested. And the other part of this is a lab assay. It's not trying to be a direct measurement. This is an indicator. It is indicative of microbial activity. So the artificial things that we do in the lab to prepare the soil, that has to be standardized in a certain way, and the amount of CO2 that comes off from that procedure is related to that microbial activity and microbial biomass. It's very different than what's happening in the field. It's very different than saying, oh, we have high respiration, so that means we must be blowing off a lot of carbon in the field. It's very different. This is a relationship here between that microbial biomass and the respiration test. Just give me a little data back up here. These are response ratios. They played a huge part in our understanding for how these different indicators relate to management practices. These are two different forms of respiration. Cornell uses a 96-hour test rather than the 24-hour test, which is, I'd say, more common in labs now. Because I didn't put any description on here, I want to tell you what a response ratio is. Basically, we had paired treatments. And so the only difference between treatments that went into here is the number of crops in the rotation. The only difference between treatments in here, so these are paired treatments. The only difference is one of them, they removed the residue, and one of them, they kept them. And same thing over here. There's either crops or there's no crops. And over here, we use the stir rating with soil tillage intensity rating to say, is it more intense tillage or less intense tillage? These are the number of paired treatments in each of them. And what you see is that residue retention, organic nutrients, cover crops, and decreased tillage all increase that respiration. Epoxy. I do want to spend just 1 minute on this. This is something that is not part of the Soil Health Institute's minimum suite of soil health measurements, largely because at the time we were doing it, we didn't have a really good interpretation. It was an award-winning prize from people at OSU. I was working on this project with them where we really focused on understanding what it was that epoxy was oxidizing. So it's this permanganate chemical, and it oxidizes a certain portion of carbon. And for a long time, we called this active carbon. We thought about it as the readily available microbial food. But really what they found in here is that it's the polyphenols, it's the aromatics, it's the tannins, it's these things that we would historically have thought of as recalcitrant type materials. But there's some really interesting relationships to microbial signaling molecules. And again, nerding out on soil science, we can do that anytime you want. I think that it is a really useful thing in here because it gets to this really high relationship to overall soil health and the microbial communication signals. I'm not going to focus on this except for to say that this is disease suppressive soils versus disease receptive soils, I guess you want to say. And there's a big difference in that epoxy distribution between healthy potatoes and diseased potato fields. Epoxy has the highest overall correlation to this overall soil health score. It's higher than organic carbon. there's something going on here that I think is really interesting, and we're still digging into it.
And I think one of the interesting points about that, which I love to reiterate, is that we thought epoxy was this thing for the longest time, and then Marcus and his graduate student and somebody else were like, no, this is inaccurate. There's a mechanistic approach to figuring this out. Point being that there are indicators that are evolving as we discuss this. I mean, that came out like last year. So
yeah. And I would say half of the soil carbon scientists don't even know this yet, right? Like this is new information. And yes, it is an influential paper, and there are a lot of people who are starting to pay attention to it, but like I will tell soil health scientists this who have been focused on soil health for the last 20 years and they've never even heard of it before. So this is new, this is evolving, this is an active area of research. Aggregate stability. Basically, we're just trying to determine whether or not particles stick together when there's some kind of force of water that is exerted. There's a lot of different ways to do this. It is time consuming. It is expensive in the lab, except for using the Slakes method, which is an app that you can get on your phone. You can do the Slakes method on your phone. It works relatively well. The only thing I'll say on here as far as interpretation is don't use it if there's more than 60% sand, because those sand particles are just going to sit there and they're not going to change, and it's not going to be very reflective of actual aggregate stability. This is, again, just some data from that NAPE shipping project. Using each of these different methods, you see the same kind of pattern where row crops have the lowest aggregate stability, and then integrated row crops and perennials are in the middle, and then these grass perennials tend to have the highest. Same story, but different absolute values based on the different method that is used. There's a video here of looking at the Slakes app. We're not doing that. Sorry, I'm skipping that. Multiple measures are redundant. These are 4 different nitrogen indicators that we measured, looking at, again, these response ratios. So don't worry too much about what the numbers are. The point is that each of them tell the same story. So we don't need to measure all 4 of them. And rather, what I would encourage people to do is to take more samples and measure fewer indicators. There is a time and a place for a lot of different kinds of measurements, absolutely, but a lot of people are kind of coming to, let's use a minimum suite here, right? So we've got, aggregate stability, active carbon, respiration, total carbon as kind of that minimum suite. pH is really important because it is one of those inherent drivers of everything that's going on. But again, you can manage it. It's inherent, and pH is a longer story. There's one other indicator that I want to talk about that's not part of the minimum suite right now, but I think there's a lot of really interesting promise into it right now, and that is using mid-infrared spectroscopy. It is quick, it's high throughput, it's non-destructive, it's kind of this like fingerprint of the chemical, organic, and mineral composition in the soil. And so Nate and I are both on a proposal where we're working on doing this in a lot of areas in the Northwest. I think there's a lot of really exciting stuff to do in here. I also think there's a lot of really exciting stuff to do with volatile organic compounds and the microbial community. But again, this is evolving science. So we're not there to set that as a minimum yet.
And that's a partnership too, just as an FYI, with the folks who did the Washington Soil Health Initiative and the WSDA. So there's regional like collaboration on trying to figure these things out.
Yeah. So summary for the minimum suite of indicators. I'm putting this slide up again. That minimum suite that the Soil Health Institute recommends and is pretty similar. I'm going to add epoxy to this, but total organic carbon, respiration, and aggregate stability as an absolute minimum to understand the total food that's there, whether the microbes are using it, and then whether the microbes are actually doing something useful with that with the carbon. Again, the goal is to get to these outcomes in context so that we can think about something like the rate that the water is moving through the soil as an indicator or as an outcome. We talked a little bit about these outcomes of interest. This is the crux of, I think, what is different in our database, and what we're trying to do is we are trying to be able to connect these indicators to the outcomes to get rid of that old circular logic. We want to make sure that we can say, okay, this indicator actually is reflective of something that the producers care about. And the way that we're doing that is by building this database. So I'm calling it SHOCKED, the Soil Health Outcomes Collaborative Database. And a huge part of it is this coordination among a lot of different people that are working on a lot of different projects. As a statewide specialist, there is no way that I can measure outcomes on all of these different soil types and all these different production systems with all of these different related outcomes. There's just no chance. And that's why we need this network. That's why we need to have this hub. We need to be able to to have different people working on different priorities for their areas who say, okay, this is what we need to do. This is the outcome that my producers are caring about. Here's where we're going to start. My job is to coordinate that so that we're using similar measurements, so that we're using similar measurements in the field and in the lab, and so that all of the management history can have a similar format, so that we can put it all into a database, so that we can start making inferences on how these producers can connect their management to the outcomes. We want to be able to connect the management to the outcomes and be able to give them some measurements that are clear that this indicator actually says something about the soil's ability to function. People are going to be able to participate in two ways, either just doing a minimum data set. Maybe it's a producer, they're not ready to like do an extensive experiment, but they want to be able to submit samples. All they'll need to do is submit a minimum management, and they're just going to get minimum indicator values, and that will help build the database. For research partners, we're going to be thinking about this outcome-based database where we have more detailed management history. We're going to take a lot more soil health indicators, including mid-infrared spectroscopy and volatile organic compounds and microbial community. And we're going to measure the outcomes so that we make sure that we've got that link between the management, various different indicators to make sure we're finding the ones that are most indicative of the soil's ability to function, and then also having that management, that measured outcome. The goal is to be able to create an interpretation tool where we are within a soil type, within a region, within a production system. We understand where we are along that soil health journey. I want to make sure that everybody can email me. Anybody can email me and ask me any other things that you want to know. I see a huge need for having a presentation on actually doing the field methods, as well as presentation on plant characterization for use in cover crops or intercrops or perennials, so that it's kind of that, okay, we go beyond the principles to saying, what plants can I actually get in the ground that are going to help my system with these functions, and how does it influence the soil's ability to function? So I'd like to do one again on that later. It will be a next year thing. Sage wrap up?
Thank you. Yeah, but we have some time for questions. And if you can go back just one more slide to the database, what is kind of that call to action to this group here? Would you like them to optionally reach out to their producers they're working with to either join the minimum data set or outcome-based data set if sampling is within y'all's capacity? Or yeah, can you kind of share that call to action to this group?
Sure. So as far as call to action, I would say this right here is the start of it, right? This is building that network of having that common understanding so that we're using common practices. And what I am asking for you to do is to, if you have a research project, reach out to me. Let's make sure that, I can help you with protocols. I can help you with methods. We can make sure that whatever you're doing is going to be able to inform this database. And maybe that is working with producers and just doing regular soil health sampling and getting that minimum management and minimum indicators. I would expect most people are somewhere in there. Anybody who wants to do a research project, anybody who says, I'm having a really hard time with this production system or with this soil type or whatever it is, let's talk about what a research project would look like, and we can work on putting something together at that small regional scale. We can do lots of small projects that are coordinated to help feed a larger overall goal. You probably know that there's a lot of challenges with funding science right now. But there are local commissions and local groups who have much more power to actually do something that is really relevant to what the producers need. And so if we have these small projects, and this is, a common theme, then we can have a lot of strength and really increase our impact.
So there's already a lot of soil testing done, including some of the soil health indicators in my area, but it's being done by seed companies. And so the information is all private. And so I'm wondering how you address this. Is there some way to keep information somehow private, or are you looking at spatial? I'm not sure how the two fit together.
Yeah, that's a really good point. Number one, there will be an absolute, a very clear data management system that will be going into this, so that there's no way to like trace back to an individual producer, or say, hey, this guy is not using any soil health management practices, we're going to go after him because he's not doing anything right. There will be and this is in the works as far as like writing up what this data management plan looks like. That's an important point. It will be taken care of. Another point that goes along with that you were talking about is the private companies that kind of hold a lot of this data right now. I have had conversations with folks at Valley Ag and Wilbur Ellis and Nutrien, and Helena Ag so far about potentially working in this soil health outcomes collaboration. I used to work for Wilbur Ellis, and they are very interested in this work and where we can go with it. They're going to have their own system to a certain extent, but they also sound like they're willing to share certain parts with us that will be able to inform the data. And so I think that, that's something to keep in mind, how that's going to work. It is still in development. But there is interest. I am absolutely talking with the private industry folks.
And then we got 2 questions in the chat. Brianna, do you want to ask yours yourself?
Just wondered, Shannon, some soil labs, really take a while to get results back. And we know that those samples are sitting in a bag, in a box, and, sometimes 6 to 8 weeks or longer. And I just wonder if, based on what we've heard here, if those results after that amount of time still have value, or if we can, is there a way that we can adjust for that kind of unexpected biological circulating or?
Yeah, and I would say, number one, it is really important when you're collecting samples not to let a moist soil sample sit on the dashboard of your truck for any period of time, right? We want to take the sample and put it in a cooler to slow that microbial activity. When they get to the lab, even if it takes a long time to get to the analysis, what they're supposed to do is air dry the samples. All of the soil health indicators are done on air dried samples. And so when we do the air drying, it doesn't stop it altogether. There is still some stuff going on, but it really slows it down. And so one of the experiments that I did when I was managing the soil health lab at OSU was doing, taking the same soil sample that has been done, dried over time, and running that POXY, that permanganate oxidizable carbon. That one does change. And it changes from lab to lab as well. Having that coordination, having that similarity within lab is also important. Along those lines, I really want to work with the soil health lab at OSU. It's where I kind of started in my soil health journey, really. Like I started managing that when the lab was about to go under. We turned it into a soil health lab. They have been doing a great job, ramping up and improving and making it more profitable so that the university doesn't shut it down. But it's really slow, and there are some challenges with it. And one of the goals is to be able to make improvements there so that we can all be sending our samples to the same lab, and having the same methods going on in the lab as well. Yeah.
Meter or those meters that can measure microbe action in real time.
Are you talking about microbiometer? Yeah. It can it has a relatively good correlation, and it's just not as standardized, really. If we're going to talk about microbial activity, doing that respiration test is more standardized. It's more national. There's a chance to use that, and like I said, there's a lot of different things you can do in the field. It can help give an indication. Absolutely. The SHAPE project, I would say, is not. It's not so different, they're working on their ways of soil health sample, grouping and analysis of these features. But again, with SHAPE and with a lot of the other projects, they're not necessarily focused on that tie between making sure that the indicators are actually saying something about the soil's ability to function, about those outcomes that are going to change producers' hearts and minds. And that's, you know, we know certain ways of how to increase soil health. We want to be able to make sure that the farmers are seeing the why they're going to do it and why they're going to deal with this long wait or reduced yields or changing markets or whatever else.
And I think that goes to Rihanna's point about the microbiometer too. Even if that there's nothing functionally wrong or mechanistically wrong with the way that it's doing it, there's still not enough information on one sample or the 10 or whatever they have to correlate it with anything at this point. And I know that they're building that type of thing, but to me, and there's nothing wrong with pursuing another line of inquiry or whatever it might be, but if you're a grower and you all work with growers and they're asking about spending money on something, like I just don't know what the value is there. Again, I'm not saying don't do it, but sort of I am. But they're like, what does that mean for that grower? What information are you getting from it? I think that's what a big part of what Shannon's talking about is linking these things to an actual outcome, whatever that outcome of interest might be.
Mm-hmm.
All so much for joining today, and thank you so much, Shannon and Nate, for a great in-depth presentation. Thank you all.